Hello, everyone. My name is Lynn Boxer, and I'm the team leader for the Cell and Tissue Products team in the Division of Animal Bioengineering and Cellular Therapies in the Office of New Animal Drug Evaluation. I want to welcome you to CVM's webinar. We'll provide you with an overview of the recently issued draft guidance documents on good manufacturing practices and donor eligibility for animal cells, tissues, and cell and tissue-based products. This webinar is primarily geared toward sponsors and developers, although it's open to everyone. This webinar consists of 3 videos. This is the first video, which will cover opening remarks and regulatory and policy framework. The second video will cover good manufacturing practices, and the third video will cover donor eligibility for animal cells, tissues, and cell and tissue-based products. Thank you again for joining us. I will now turn it over to Dr. Stephen Solomon, who will offer some opening remarks.
Thank you, Dr. Boxer. Hello, everyone. I'm Steve Solomon, Director of FDA Center for Veterinary Medicine, or CVM. We're very pleased you could join us for FDA's webinar about good manufacturing practices and donor eligibility for animal cells, tissue, and cell and tissue-based products. We refer to these products in shorthand as ACTPs. Here at CVM, our mission is to protect human and animal health. Meeting this mission is what motivates CVM staff who are deeply committed to public health and passionate about animal health. One of the ways we accomplish our mission is by encouraging the development of innovative products such as ACTPs. ACTPs have the potential to make significant impact on how we treat a variety of disease conditions. Especially exciting is the opportunity they may provide as novel therapies for unmet therapeutic needs for animals. While we strongly encourage development of ACTPs, we make sure these products are safe and that they perform as expected. This is critical to protecting human and animal health, as well as to fostering consumer confidence in these novel products. We use a risk-based, product-specific approach to regulate ACTPs, and we're committed to making regulatory decisions about benefit and risk based on sound scientific and technical evidence. We recognize that the science and technology around ACTPs are rapidly evolving, and that these are unique considerations associated with manufacturing them. These include how to recover, process, store, label, package, and distribute them, as well as how to administer ACTPs. Critical concerns for ACTPs included the potential to transmit disease between donors and recipients, as well as preventing contamination and maintaining product quality during manufacturing. We understand that these products pose unique risks and must be properly evaluated to ensure safety, effectiveness, and quality. We're committed to advancing development of these innovative products in the most streamlined and efficient manner possible. As part of this commitment, we recently issued 2 draft guidance documents for ACTPs, one on good manufacturing practices and another on donor eligibility. These draft guidance documents are intended to assist manufacturers by providing product-specific recommendations that will help developers meet existing FDA manufacturing requirements. We also encourage developers to take advantage of the Veterinary Innovation Program, or the VIP program, which is aimed at encouraging development of innovative products by assisting developers in generating the appropriate data needed to support approval. Our goal is to help developers be successful in their efforts to bring to market safe and effective products that benefit animal health. We hope this webinar will provide you with an understanding of how good manufacturing practices can be applied to the manufacture of ACTPs to prevent contamination and ensure quality. We also hope you'll gain an understanding of FDA's risk-based recommendations for selecting appropriate donors and preventing transmission of disease between donors and recipients. Thank you again for tuning in to our webinar, and I'll turn it back to Dr. Boxer.
Thank you, Dr. Solomon. Let's take a look at the webinar agenda. We'll start with a short overview of the laws, regulations, and guidance applicable to ACTPs. Next, we will discuss the two new draft guidance documents recently issued by CVM. We'll focus on the concepts presented in the draft guidance documents 253 and 254, including product-specific manufacturing considerations and donor eligibility. These draft guidance documents provide FDA's recommendations and are intended to assist developers of ACTPs in complying with current good manufacturing practice requirements. If you have questions after the webinar, you may submit your questions to our FDA CVM Animal Biotechnology Feedback email. We will respond to your questions through individual communications and publication of additional outreach materials. Before we discuss the draft guidance documents, I want to take a minute to talk about CVM. CVM is charged with ensuring the safety and effectiveness of animal drugs. Fostering innovation is one way that we support development of novel, safe, and effective products for use in animals. We coordinate and collaborate with experts across the agency to leverage knowledge that furthers development of both human and animal cell and tissue-based products and promotes consistency in the regulation of these products. We also conduct research to address scientific challenges facing ACTPs and to facilitate efficient review of ACTPs. We develop resources to assist veterinarians, owners, and developers, such as our web pages listing clinical field studies for ACTPs and lower risk ACTPs. We also conduct outreach and implement programs such as the Veterinary Innovation Program, which is specifically designed to assist developers of ACTPs with the approval process. In addition to these center-wide initiatives, in the Office of New Animal Drug Evaluation, or ONAID, we formed the Division of Animal Bioengineering and Cellular Therapies to evaluate emerging technology products such as ACTPs. This division has critical expertise across a wide array of scientific disciplines, including cell and molecular biology, veterinary medicine, bioinformatics, genetics and genomics, chemistry, and animal science. This division provides scientific and risk-based oversight, ensuring safety and effectiveness of ACTPs and other emerging technology products. This division works hand in hand with other divisions in ONAID. Other divisions in ONAID, including the Division of Manufacturing Technologies, also provide critical expertise to the review and regulation of ACTPs. During today's webinar, you will hear from members of the Division of Animal Bioengineering and Cellular Therapies and the Division of Manufacturing Technologies. Today's webinar is focused on the two recently issued draft guidance documents. When FDA issues a guidance in draft, there's a public comment period is provided in the notices of availability published in the Federal Register. You may submit comments on the draft guidance documents to the docket numbers provided at the links shown on the slide. While you may submit comments on guidance documents at any time, we are requesting that you submit comments by November 22nd so that we can consider those comments when drafting the final guidance documents. To submit comments, go to www.regulations.gov and enter into the search box the docket numbers provided on the slide. Alternatively, you can type the link shown on the slide directly into your browser. Please note that sending us an email is not the same as submitting a comment to the docket for consideration in drafting the final guidance documents. All comments on the draft guidance documents should be submitted to the docket. I will now hand it over to Dr. Janet Mendonca, a cell biologist on the Cell and Tissue Products team in the Division of Animal Bioengineering and Cellular Therapies.
Thank you, Dr. Boxer. I will now discuss the statutory, regulatory, and policy framework for ACTPs. ACTPs are articles consisting of or derived from cells or tissues that are intended for implantation, transplantation, infusion, or transfer into an animal recipient. This definition covers a broad range of biologically derived products. Generally, ACTPs are products that meet the statutory definition of a new animal drug. These products are regulated by the FDA under the authority of the Federal Food, Drug, and Cosmetic Act. ACTPs generally meet the definition of a new animal drug if they are intended to diagnose, cure, mitigate, treat, or prevent disease in animals, or are intended to affect the structure or any function of the animal. As new animal drugs, ACTPs are subject to the same statutory and regulatory requirements as other new animal drugs. Examples of ACTPs may include mesenchymal stem cells, exosomes, placental tissue, pancreatic islets, bone marrow, blood transfusion products such as whole blood and blood components, blood-derived products such as platelet-rich plasma, and tissues such as cornea and bone. The Federal Food, Drug, and Cosmetic Act, or FD&C Act, is the law or statute that provides FDA with the authority to regulate new animal drugs. The law requires new animal drugs, including ACTPs, to have an approved or conditionally approved new animal drug application or index listing prior to marketing. The law also requires new animal drugs, including ACTPs, to be manufactured in accordance with Current Good Manufacturing Practice, or CGMP, to ensure that such drugs meet the requirements for safety and have the identity, strength, quality, and purity characteristics that they purport to or are represented to possess. This aspect of the law is referred to as statutory CGMP. Regulations have the effect of law and explain how the law is implemented. The Code of Federal Regulations, or CFR, provides legally binding requirements regarding approval, investigational use, manufacturing, and other aspects related to the regulation of new animal drugs. The CGMP regulations for drug products are found in the Code of Federal Regulations in 21 CFR Parts 210 and 211 and are referred to as regulatory CGMP. These regulations provide specific requirements for the organization, personnel, facilities, equipment, distribution, controls, and records used in the manufacture of new animal drugs. All new animal drugs, including ACTPs, are required to be manufactured in accordance with CGMP. While regulatory CGMP is written broadly to cover all types of new animal drugs, FDA recognizes that the regulations do not specifically or fully address all aspects of the manufacture of ACTPs. For example, the regulatory CGMP does not address the early stages of the manufacturing process or donor eligibility. While the law and regulations may not specifically address unique aspects of the manufacture of ACTPs, the broad nature of the law and regulations provide FDA with the authority to ensure the identity, strength, quality, and purity of an ACTP. New animal drugs not manufactured in conformity with statutory and regulatory CGMP are adulterated under the relevant provisions of the FD&C Act. Guidance documents represent FDA's current thinking on a topic and are used to provide non-binding product-specific recommendations. For example, a guidance document may provide FDA's recommendations on how to comply with an existing law, such as the statutory requirement in the FD&C Act, or a regulation, such as the regulatory requirement in the CFR. Guidance documents are not legally binding on FDA or the public. Alternative approaches can be used if the approach satisfies the requirements of the applicable laws and regulations. In 2015, FDA issued Final Guidance 218, Cell-Based Products for Animal Use, to provide clarity on how existing laws and regulations for new animal drugs apply to ACTPs. Guidance 218 also provides information on investigational use and considerations for approval. FDA recently issued draft guidance documents 253 and 254 to provide recommendations for those aspects of manufacturing specific to ACTPs and to assist manufacturers in complying with CGMP. Draft guidance documents 253 and 254 were issued to assist developers of ACTPs in complying with CGMP. These documents take into account the unique considerations of sourcing and manufacturing ACTPs. The draft guidance documents make product-specific recommendations intended to help manufacturers of ACTPs ensure product safety and comply with existing CGMP requirements for new animal drugs. Draft guidance document 253 provides recommendations to help establishments that manufacture ACTPs meet statutory and applicable regulatory CGMP. The draft guidance document addresses the methods, facilities, and controls used for manufacturing ACTPs, including steps in recovery, processing, storage, labeling, packaging, and distribution. Manufacturing in accordance with CGMP is critical to ensure that the ACTP meets the requirements for safety, effectiveness, and quality. Draft guidance document 254 provides recommendations for complying with CGMP for those aspects of manufacturing related to donor eligibility, including donor screening and testing. Donor eligibility is critical to preventing transmission of relevant disease agents and diseases and ensuring the safety and quality of allogeneic and xenogenic ACTPs. In today's webinar, we will provide examples to demonstrate practical application of the concepts in these draft guidance documents. The case scenarios and examples discussed in this webinar are intended only to demonstrate individual concepts. The case scenarios and examples may not provide the complete information needed to address a specific consideration and may not be applicable to all ACTPs or all donor and manufacturing scenarios. This concludes the first section of our webinar on good manufacturing practices and donor eligibility for ACTPs. In the second video, we will discuss draft guidance document 253 in more detail.